Hi, everyone. I think we're about 35 minutes past the hour right now. So if you don't mind taking a seat, we can get started with our third and final session for today. I know. Thanks. One quick announcement right now. If you drove here today and parked in the garage that's attached to the Bethesdan, we do have vouchers that will take down the cost for parking to the day to $10. So if you would like one of those vouchers, go find Laura Lummis at the registration table and she will have one for you. Thanks. All right, we are here at the third session for today's workshop, and this one we're looking at the biological performance considerations, and really what we're focusing here is what are the necessary study performance attributes, that are related to the specified context of use. And so we'll have two presentations here, and then move into panel discussion similar to the first two sessions. So I'd like to introduce first Ravi Kodihalli. And Ravi is a senior principal scientist at Pfizer in their drug safety R&D division, and is going to talk about his perspective around development of a context of use and appropriate biological performance testing of that.
Thank you, Nick, for kind introduction. Thanks to Graham and Katrina and all the organizing committee for giving me the opportunity to talk about industry perspective on the development of context of use for liver complex in vitro models. More than 50 years back, Russell and Birch wrote a book on the principles of human-animal research. In that book, they alluded to an idea of 3 R's: replacement, reduction, and refinement for performing animal research. Since then, many national and international agencies come up with their policies and regulations around those principles to perform animal research. As a pharma, we try to follow those principles, and by utilizing the 2D models, 2D in vitro models, and the complex in vitro models, and in our drug discovery settings. Whenever it's possible, we try to make decisions on the data we used to get it from those CIVM models. And if needed, we try to reduce the number of animals and try to make a decision on the data from the complex in vitro models. As this survey indicates, I think it's kind of an advantage for us, because as the survey indicates, we can save nearly 10 to 26% in our R&D cost. But the effective implementation of these complex in vitro models needs well-defined context of use from the regulatory agencies. Then only it's possible for us to refine or distill the entire drug discovery process. We think the context of use as minimum 3 components. One is an engineering aspect of the microphysiological system to achieve the desired objectives. For example, anatomic and physiological specification for a given study, and a degree of non-specific binding. And the model, the engineer tools have the at least minimum robustness. Also, it should hold the number of replicates as well as the number of donors. It has to be characterized on those engineered tools. And finally, there are some functional and practical consideration in terms of achieving the end point. In terms of an organ-specific model, yes, we do want an COU around testing the toxicity. And also, since we are talking about the liver complex in vitro models, we should know what are the ADME genes should be present in those models, and what kind of CYP enzymes, what kind of transporter should be active in those systems, and what are the cell types we are going to culture in those organ systems, and also number of cells, and also those systems should susceptible to the sampling, frequent sampling, and also within that number of cells, we are able to achieve the desired endpoint in terms of a biomarker or any endpoints. Finally, the regulatory requirements, we need to have a specific guidance in terms of SOP, standard operating protocols, for any CIVM models, also the standardized data processing protocol and interpretation methodology we need. Also, we would like to hear from them is that what stage of the development we can apply these complex in vitro models. Also, what is the species relevance? So it's very important to first to establish the fit for purpose qualification parameters to recapitulate the entire human physiology, to understand the drug response as well as the disease development and the progression. So there are many hurdles, I think, in utilizing these complex in vitro models in the drug development, because cells needs in the structure of scaffolds, and those need, I think the extracellular matrix, I think different matrix have a different chemistries associated with it, because they need a proper they need to create a proper architecture, as well as they need to give some kind of polarity, as well as they need to survive in the system. And so far what we heard is that there are some production related issues with those matrices so far. And if you think about a platform, now we are thinking about only the liver complex in vitro model. What if down the line, if we want to integrate into the other in vitro models, if you want to understand toxicity between the liver and the kidney, or GI and the liver. So these models are a platform when we are considering for the qualification, and also those are many challenges we're facing. Those should be having like a plug and play methodology. Whenever it needs, okay, we want to test in liver, test it in liver system, we can test it. And if you want to connect the kidney or other organs, we need to have that kind of capabilities. So far, only few studies demonstrated that from some of the DARPA projects. And the cell sourcing, this is another challenge we always face, because primary cells isolated from the liver biopsies have a minute in amount, and those are functionally and physiologically mature. And those units, some of the units currently available in the market they need an expertise to handle those tools. It's not simple as 2D systems, simply add hepatocytes and test things. There are challenges associated with that. Irrespective of those challenges, we saw a lot of bright light in these in vitro models. There are tremendous applications. So far in the market, every organ systems are available, and cell types also we can test on those models. And currently we're using for the safety testing purpose and disease modeling purpose, and understand the ADME potential, as well as some cases we try to understand, we need to make internal decision making using the data we used to get it from the complex in vitro models. I think the data can be provided for the regulator submission whenever the regulator is to ask. We think the context of use has to be defined by the FDA based guidelines, biomarkers, endpoints, and other tools where medical products are used for the drug development purposes. So when we are thinking about the developing context of use, we should think about the translational axis to address some of the complex problems in the drug discovery process. So far, from morning session, Jeff and Mark talked about on this side of the qualification, especially in the clinical settings. I think I do agree. I think there are, they show the feasibility, especially they can mimic the pathophysiology of drug responses that was observed in the humans but not detected in the animal models. For example, human blood vessel microphysiological system able to identify the thrombotic toxicity. Those are treated with the monoclonal antibodies. Another example is that few years back, proximal kidney tubule models that are able to identify the cisplatin metabolite that was metabolized by the PGP transporter that only present in human but not in the animals. So also when we are performing, when we think about the context of use, we need to think about the bottom approach too, because since I came from the drug safety R&D, we think this is also very critical, because always when we are testing these drugs in the preclinical setting, we used to use these complex in vitro models. When we are performing the animal studies, we can parallelly conduct the studies along with the animal studies, because a lot of these models are available for the preclinical use as well. So that we can collect the data parallelly along with our preclinical studies. So during that time, we can use the data for the regulatory submission, and also we can collect the data for the other endpoints such as biomarkers, so for example, proof of concept, as well as the small and large molecule data, as well as traditional markers. So you can take it. Sorry. So, and also now the statistical tools are available to integrate the data. So in the entire process, we are going to collect the huge amount of data, and these statistical tools as well as the biomarkers act as a bridge for the entire translation, because these tools have become after a translation tool for the entire translation tools. Because now we are only thinking, if you think about only on this side, in the down the line after 2 or 3 years, once we started developing molecules, when they go back to the clinic, if something happened here, then we can come back and look back into the data set which is already generated, it's already in-house. So in the entire process, as I mentioned, we are collecting a huge amount of data. Then we can think about a data integration strategy, because from the preclinical data, CIVM data, and historical data, that's from data coming from the clinic, then we can use the artificial intelligence, machine learning, deep learning tools to make a data-driven decisions and come up with an high-quality targets. Because this is like drug-induced liver injury. I think that's a very complex that needs a multi-parametric endpoints and multi-parametric approach. So these AML tools and the data integration, effective data integration strategies will help us to understand and to identify any risk associated with these molecules what we're planning to take into the clinic or from the clinic as well, due to daily risk. Since we are talking about the complex in vitro models, I would like to shed a light on the currently available models, what we are using in the pharmaceutical industries. I think this is an old paper in 2020. I think this is an update a little bit. And here the 2D models are not there. When you think about all these complex in vitro models, I think you can see in the last column, there is a context of use. For example, for safety assessment, I think all the models will fit better. And if you understand if you understand immune-mediated delay, then stem cell-derived organoids will be better models. And if you think about the future, and liver chip models will be there. Those are the best. That's the way we need to move forward, because if you want to integrate in the future other organs, I think we need to consider the liver chip models, so that we can add the monocultures and co-cultures and multiple cell types from the different organs too. We can connect it and we can test the toxicity of those drugs. So as it indicates, this very important thing is that for us, is the high throughput and the reproducibility and the cost of these tools are critical, because the reason we are using these in vitro models is to get a quick answer and to reduce our R&D cost. I think when we are developing this context of use, we need to make sure all these things will be all these things are for consideration. Then the toolkit. I think last talk Kevin mentioned about the list here that's presented in 2020 from the IQ consortium companies. As you can see, I think here's another paper. They screened around 160 molecules, and out of that, they presented data for the around 40 based on the ToxSpy scores. And as you can see, from the increase in the key percent of, I think y-axis is the percent of live cells. I think I think safe drugs to the decrease in the, I think, increase in the toxicity here. And also these are some of the reference compounds. We won't recommend, I think you can go with these molecules. I think these acts as a toolkits. So, and also if you need a more compounds, I think if some compounds are IP protected from the companies, then we can use some of their derivatives for the testing purpose. Then we can create a pool of around 40 to 50 compounds, then try to use those compounds for the validation purpose. So far, in terms of a complex in vitro models in the pharma settings, I think they worked really well for us so far, and we're using many of the complex in vitro models for the selection of best candidates, as well as some of the early validation purpose and identifying some of the molecular mechanisms, and also understand the safety liabilities of those compounds in the preclinical setting itself. And in some cases, we use that data for the selection of doses for the preclinical studies. Also, in the clinic, yes, there will be we can use these tools for the human risk assessment. But the one limitation is that the guidance from the regulators, how to use these models, how to interpret the data, and what context we need to use. And still, we don't know what MPS models or CIVM models will be better in terms of for regulatory submission. That's one of the things we need to consider. And finally, the cost and the throughputs are the main important criteria. So in summary, we are here to discuss about the liver complex in vitro model. As I mentioned previously, it's a complex. Deal is a complex. I don't think so one solution would fit for all purpose. So it needs a multi-parametric approach. Then only it's possible to make a data-driven strategies to find an effective solution. Then the question is that who is responsible for it, right? Because academicians develop a great models, publish a great publications. Then the developers optimize the models, and they will commercialize these products. Pharma always excited to use the new tools if they are able to, if you give robustness and if it's less cost, we are always excited to use those in vitro models. And also the regulators, I think morning Jeff mentioned on the iStand, I think there are some initiatives already starting, they're doing it. So all these things will be helpful. If you can work, all of us work together, then only it's possible to bring the safe medicine in a light speed. With that, I would like to acknowledge Jonathan Jackson is here, and online Dr. Ania Kopek and Tom Lance, and also our executive team for their support. Thank you.
Thank you, Ravi. So for our final presentation today, we have Qiang Shi from the FDA at the National Center for Toxicological Research, where he is a senior staff fellow. And he's going to give a perspective from his research lab around biomarkers and meaningful endpoints.
Hello, everyone. Yeah, thank you, Nick, for the introduction. I know I'm the last speaker. We had a long day. I will try to brief. Can I say a big thank you to those who already left, because you guys made me feel less nervous. So, I'm working on two projects studying drug-induced liver injury using various commercial liver chip platforms. So in this talk, I'm going to share some of our experience or lessons that we learned during this progress. I want to go back. Sorry about that. I'll go back to this one. Oh, thank you. Sorry. So the presentation will focus on liver CIVMs. Of course, we know it's most advanced and mature ones, and it's widely used in drug metabolism and drug-drug interactions, and of course, drug-induced liver injury. And, I think probably most of us noticed that primary hepatocyte are the recommended model in the couple of FDA guidance for industry documents. So I guess that's the reason primary hepatocytes are considered as a gold standard for drug metabolism and drug-drug interaction studies. That is why we choose to use this model as I think Evan Ford mentioned there are lots of cell lines available, but we choose to use the primary hepatocyte. And actually, findings based on primary hepatocyte have been included in FDA-approved drug labels. Here's an example. So regardless of I know we had lots of discussions of COU, but I feel that regardless of COU, there are some biomarkers that are needed to determine if the cells are alive or functioning. This is really important for primary hepatocyte because the major challenge for hepatocyte, primary hepatocyte is the spontaneously and time-dependent loss of cell viability and function. And so in our study, we thought that biomarkers for CIVMs and for regulatory COUs for CIVMs could have similarities and some differences. And I do want to say that these new models they were developed to address the drawbacks of traditional cell culture. So it's not necessarily for any defined regulatory COUs. I guess that's probably the reason that we're kind of struggling here. We're having this meeting here. And in my talk, I'm going to focus on markers that could potentially be used for both purposes. So we know there are numerous reported markers, biomarkers, endpoints that could be used for these systems, but only a few clinical used biomarkers are being used by FDA for safety assessment. So I'm referring to the FDA guidance for industry regarding daily. So, and in the literature, many biomarkers have been proposed for CIVMs, but the translation to clinical relevance really isn't very clear. So the goal of this project is a qualification of selected CIVMs for defined COU in a regulatory assessment. So here's the kind of the priority levels. We decided we wanted to first focus on the markers that are included in FDA guidance for industry for drug-induced liver injury. And the second one category, we want to focus on biomarkers that are submitted by CFAS for FDA and EMA qualification. And the third one are the markers that are included in the clinical guidance. And the fourth one is guideline papers for the liver MPS. The last one is research articles. Yeah, I apologize for those who came from academia. So here are some general considerations. So ideally, we believe that the markers should reflect the dynamics of in vivo biomarkers, but this is really not always possible. As we know that bilirubin is FDA used daily biomarker, but it's not applicable to the CIVMs because it is a metabolite of hemoglobin, and I don't think any CIVMs currently have red blood cells in their systems. However, liver CIVM biomarkers may indeed have their own unique advantages, so they may help address the drawbacks of standard in vivo biomarkers. One example is that we heard a lot that ALT and AST, these clinical use well-established biomarkers, they are not specific to liver injury, because muscle injury may also lead to their elevations. So we feel that if we use the CIVMs of both liver and muscle cells, this may help us differentiate if the ALT and the AST elevations are mainly from liver or non-liver cells. So here's the group 1 biomarkers. So these are the markers described in the FDA daily guidance and another draft guidance that was recently released. So in these guidances, it's the 4 categories of markers are included. First 2 are well-established, the ALT and the AST. They actually reflect cell death. So they are cytosolic enzymes that are released when cell death occurred. So it reflects cell hepatocyte necrosis. And another one is ALP. It reflects cholestasis. And the third one, it's probably very hard to measure. I don't think any CIVM models measured this marker. It's vitamin K dependent clotting factors, but in traditional 2D cultured cells, I indeed saw some literature report about this. I believe probably this is a good research direction. The last one is bilirubin is not applicable to CIVM. So group 2 biomarkers, these are the biomarkers submitted by CPAS to FDA and EMA qualification. Probably the best one is the GLDH. It's highly enriched in the liver mitochondria, and it's released upon cell death. The advantage of GLDH compared to ALT and AST is that they are more liver specific. So the third ones are the clinical guidance for liver diseases. So the mode for end-stage liver diseases, I think is a very interesting one. It's being long used by the United Network for Organ Sharing Systems to rank the patient on the wait list for liver transplantation. So in this model, there are a couple of endpoints included, such as bilirubin, we already talked about that, and creatinine, but creatinine is mainly a liver. It's a biomarker for kidney function, so it's not good candidate for liver. And also INR and the sodium. I'm not sure, probably sodium is also not applicable to the in vitro systems. And last one is the albumin. This one has been widely used in liver CIVM in the literature. Group 4 are the guideline papers. There are actually a couple of guideline papers. I know some speakers mentioned the guidelines paper from industry group, but there are also some other papers from academia. So it's recommended that albumin, urea, and drug metabolism enzyme transporters be used as markers for MPS. The last one, this included lots of markers, so I won't go into the details, but for the last one, I indeed want to see a couple of things. So these are the biomarkers reflecting the functions of non-parenchymal cells, including the cover cells. We know that these cover cells can be activated during the immune response related to drug-induced liver injury. So indeed, the cytokines can be measured in these systems. this table kind of summarized what I just mentioned. So we can see here the red ones, they are the markers on the in vivo studies, and the blue ones, they represent some markers, possible useful markers for CIVM models. Yeah, so here I want to share some of our experience in measuring these markers. The first one is ALT and AST. We know in the clinical ALT and AST is measured by their activities, but the challenge here is that the matrix really is different from the clinical practice, because in clinical practice, the markers are measured in serum, but in this complex models, it's measured in the cell culture medium. The matrix is cell culture medium. And to our surprise, we noticed that in this figure, it shows that the ALT activity is really unstable during the culture, in the culture system. So their activities decreased almost by half every 24 hours. In contrast, the AST activity is much stable. So this is in contrast in clinical samples. It's well established that ALT and AST activities are stable in human serum. So we believe this is a very important finding, because ALT activity has been used to assess the performance of the models. And if we rely only on this marker, we will underestimate the spontaneous cell death, or underestimate the toxicity. So here's something that's also interesting. We believe that the matrix can affect the assay sensitivity and the specificity, or the compatibility of the detection method. So in these two figures, so this one here, we measured the ALT activities after drug treatment on different days. But in this figure, we measured the ALT protein levels. And the results are from the same samples, exactly the same set of samples. We can see the big difference here. So basically, our conclusion is that ALT activity, even after drug treatment, when massive cell death occurred, we were not able to see significant elevation of ALT activities. However, we indeed see significant elevation of ALT when we measure it using the ELISA-based method. This is measuring the protein levels. So this is in line with our first slide showing that ALT activity is not stable in the system. That's one likely reason. Another likely reason is that cell numbers are just too low, so that the activities are not measurable. So we indeed, if we look at the literature, there are some publications showing that ALT activity was indeed elevated, but I seriously have doubts about such results. I'm not so sure if they are really reproducible. This is another example of the functional markers, albumin and urea. We know it's proposed that albumin level should be over certain levels, urea level should be over certain levels over time, and this is one data set from our recent experiment. So, in short, the red lines represents the albumin level, and the dotted red line represents the proposed threshold of acceptable performance. So we can see that albumin can be maintained for around 11 days here, but after 11 days, it decreased to unacceptable level. But urea is a different story. We can see, again, the green line shows the urea level measured over time, and the dotted green line shows the threshold level. It looks to me that the urea level can be easily maintained in the system, but the albumin level is kind of a different story. So we probably needed to assess the different reactions of these different markers. And we probably should also bear in mind that these suggested threshold levels are based on the in vivo production of these molecules. It's not necessarily based on the CIVM experience. So we were wondering if such criteria should be made according to in vivo levels or the experience with the cells. Also, something to consider, the dynamics of CIVM biomarkers will also depend on the system design. So we know that some systems are perfused, some are non-perfused. And for the perfused systems, we know that there are open perfusion systems and closed perfusion systems. So the open perfusion systems probably is the most widely used, but they have lots of issues because the perfusing medium was removed at desired intervals to measure the biomarker, and then we added a new medium. This could result in diluting effect. This is probably the reason we cannot measure lots of markers that's commonly used in the clinical practice. And to kind of address this issue, probably we should include mouse cells to these systems. This is a particular challenge when we treat the cells with drugs. The drugs usually will cause massive cell death, so we cannot evenly accurately measure how many cells are left in the system. So this is something to think about when we measure these endpoints in the CIVM systems. So we don't need this. This is just for the preparation of this discussion. Here are the two projects that I've been working on. I would like to thank all my co-investigators listed here, and I especially want to thank my supervisors for providing lots of suggestions during the preparation of these slides. And lastly, thank you.